Watch this brain cell's nucleus. A moment ago, it was a smooth, round sphere, held in shape by a fine protein mesh just under its surface. Now, the mesh is coming apart, and the nucleus is caving in on itself, folding and wrinkling and shrinking. The rest of the cell is still there. The first thing to fail was the vault that holds its DNA.
Biology has a catalog of ways a cell can die. There is the tidy self-destruct program most people have heard of, and there are messier routes with names like Necroptosis and Ferroptosis, each with its own trigger and its own point of no return. But the neurons lost in Alzheimer's and frontotemporal dementia never fit neatly into that catalog. A large share of them seem to be dying in some way the known categories did not fully describe. This is a candidate for a missing entry, a route where the nucleus is the first part to fail. Researchers gave it a name, Karyoptosis.
So the honest question to ask right away is the one you are probably already asking. Does this mean a treatment is coming? Not yet, and it is worth being clear about that early. What the work offers is different and still valuable, a specific physical step in how these neurons die, one that can be interrupted in the lab, and a fingerprint of that same process found in real human brains. Let me show you how they got there. The paper is from Rebecca Casterton and colleagues at King's College London and the UK Dementia Research Institute, published in Nature Communications in June 2026. Their starting point was a stubborn problem. Toxic, misfolded proteins pile up inside neurons in these diseases, and neurons are unusually resistant to the ordinary self-destruct program. So the usual explanation for how a poisoned cell dies did not obviously apply. They needed to watch the death happen. They recreated that protein clearance failure in human-derived neuroblastoma cells by jamming the cell's recycling system so junk proteins built up the way they do in disease. Then they filmed what happened over hours. The earliest clear sign of trouble was not the whole cell breaking down. It was the nuclear mesh. A structural protein called LaminB1, which normally lines the inside of the nucleus and keeps it round, pulled away from the edge and clumped into specks in the cytoplasm. The nucleus lost its smooth circular shape. And this happened before any real DNA damage showed up, about 12 hours in, well ahead of the later wreckage. That order is the whole point. In the familiar death program, the nucleus condensing is a late step in a decision the cell has already made. Here, the nucleus is where the damage starts. So what flips the switch? The team traced it to a stress-sensing enzyme called p38 MAP kinase. When the protein junk builds up, p38 turns on, and it does something direct. It chemically tags LaminB1 at a specific spot, a site called SIR391. That tag destabilizes LaminB1, so the mesh that holds the nucleus in shape starts to come apart. Here is the experiment that makes this more than a correlation. When they blocked p38 with an inhibitor, the nucleus held its shape. The LaminB1 mesh stayed in place, and the specks did not form. But the upstream protein junk was still there, just as high as before. Blocking p38 did not clean up the mess. It cut the wire between the mess and the collapsing nucleus. That is strong evidence that p38 tagging LaminB1 is the actual step that kills the nucleus, not just something happening nearby. They also checked what this death is not. Under these conditions, there was no sign of the caspase enzymes that run the classic self-destruct program, and blocking those caspases did not save the nucleus. There was no marker of the membrane rupturing route, no sign of the iron and lipid route, and blocking the DNA damage route did not help either. Different trigger, different order, different fingerprint. That is why the authors argue it deserves its own name, rather than being filed under something already known. A pathway in a dish is a start.
But does interrupting it actually protect a living nervous system? This is where the study gets more convincing. In fruit flies engineered to carry the same protein clearance failure, neurons showed the same nuclear collapse. When the researchers made LaminB impossible to tag at that key site, or turned down p38, the flies did better. Their neurons kept a healthier nuclear shape, and the animals moved better and lived longer. They saw the same rescue in rat neurons and in the fly versions of the genetic changes seen in inherited ALS and frontotemporal dementia. And in human neurons grown from stem cells, blocking p38 again protected the cells under the same stress. Then comes the part that pulls this out of the lab and into the disease. The team took postmortem brain tissue from 28 people who had died with Alzheimer's or frontotemporal dementia, plus healthy older brains for comparison. They imaged the LaminB1 and the shape of thousands of individual neurons and let a computer sort the cells into groups by their features without telling it which brain each cell came from. 2 of those groups matched the Karyoptosis signature, an early stage with the telltale cytoplasmic specks and a late stage with the collapsed nuclear shape. In the diseased frontal cortex, about 35% of cells carried those signs. In the healthy aged brains, it was around 15. Be careful how you read that number. This is not a film of neurons dying in a living patient. It is a snapshot of fixed tissue after death, sorted by a pattern the computer was taught to recognize from the lab experiments. It is a strong association, not a caught-in-the-act proof. And in the paper's own precise counts for that comparison, the diseased cells cluster around 35 to 37% against 17 in controls. A real gap, but an inferred one. There is one more honest limit worth stating plainly. Karyoptosis is not the whole story of why neurons die in dementia. In that same patient tissue, the researchers found another group of dying cells that did not carry the Karyoptosis signature at all. Some neurons are clearly going down a different road. So the claim is not that this explains dementia. It is that a real, substantial share of the loss may run through this one nucleus-first route. Which brings us back to the promise from the start. The reason this matters is that a death route you can name and interrupt is a death route you can try to drug. The specific handle here is that link between p38 and LaminB1. p38 itself is not a new target. Drugs that block it broadly have been tried and have mostly disappointed because p38 does a lot of jobs, and shutting it all down causes problems. The bet in this work is narrower and more interesting. Hit only the connection between p38 and the nuclear mesh, and you might spare the nucleus without switching off everything else p38 does. No one has done that in a person yet, but the map of how these neurons die now has a new road on it, one with a specific, testable place to intervene.